Good morning. Myself, Dr. Anil Kumar Shinde, Department of Pharmaceutics, Bharati David College of Pharmacy, Kolhapur. Today I'll review the liposomal drug release system. Learning outcome, liposomes, classification, advantages, limitations will be discussed. Realize the importance of drug release system for liposomes. Manufacturing methods, selection of drug and materials, characterization and application of liposomes, and stability of liposomes. List of content, introduction, advantages with use of liposomes in drug release system, classification, manufacturing of liposomes, liposome characterization and control, stability consideration for liposome formulation, regulatory science on liposomes, drug products, drug release from liposomes, applications, recent innovations, approved liposome product and references. Liposomes are macroscopic spheres made by fatty material, predominantly phospholipids, made up of one or more concentric lipid bilayer and range in size 50 nanometer to several micrometer in diameter. Classification based on the size: small unilamellar vesicles, medium-sized unilamellar vesicles, large unilamellar vesicles, giant unilamellar vesicles, unilamellar vesicles, oligomolecular vesicles, multilamellar large vesicles, multivesicular vesicles. Classification based on the method of preparations: vesicles prepared by extrusion method, vesicles prepared by French press, vesicles prepared by fusion, vesicles prepared by reverse phase evaporation, frozen and thaw MLV, dehydration, rehydration vesicles, stable plurimolecular vesicles. Manufacturing of liposomes: different materials can be used for preparation of liposomes, that is phospholipids, synthetic phospholipids, glycolipids, sphingolipids, glycero steroids, polymeric material, charge-inducing lipids. Issue related to consider when selecting lipids: the phase transition temperature, stability, charge, lipid mixtures, cholesterol, and sources. Conventional liposome preparation methods can be used: biosonification method, high air fragmentation, solvent injection method, injection of water miscible solvent by ether infusion, fluorocarbon injection, injection of water miscible solvent, ethanol injection, modified ethanol injection method. Different methods used to prepare LUV and IUV, detergent dialysis, water in oil emulsion technique, freeze-thaw cycling, slow swelling in non-electrolytes, dehydration followed by rehydration, dilution or dialysis of lipids and presence of glycolic ions. Methods for controlling liposome size: fractionation, centrifugation, size exclusion chromatography, homogenization, capillary pore membrane exclusion, ceramic extrusion, physical characterization of parameter for preparation of nanoparticle, bean size, size distribution, number of lamellas, osmotic behavior and trap volume, internal distribution of drug, structure and motion behavior of lipids, electrical surface potential and surface pH, stability consideration of preparation of liposomes, stabilization aspects for physical instability of liposomes, chemical stability and biological stability of liposomes. Some of the regulatory aspects required for preparation of liposomes, that is a safety concern, liposomal preparation. Lipid toxicities, that is a RBC lysis, the presence of protein and lipoprotein for natural lipids. Residual solvent, overload of RES, particle size, tail by 1 micron or blockage of capillaries. Size affect the RES uptake and tissue targeting. Stability, uh, shelf life and in dose dumping via protein binding and sterility of effluent. With this preparation of liposomes and its evaluation properties of liposomes we have discussed.